everyone. Today in our video lecture, we're talking about adaptive immunity. So hopefully prior to this video lecture, you have already learned about the anatomy of the lymph system and about the innate immunity or non-specific immunity associated with the lymph and immune systems. And now we'll dig into adaptive immunity. Adaptive immunity is also known as specific immunity because in adaptive immunity, we are going to be targeting specific antigens of pathogens. This will be done through a coordinated response by T and B cells, which are types of lymphocytes. And we're going to see that adaptive immunity exhibits four unique properties that we don't see with innate or nonspecific immunity. So those four properties will include specificity, versatility, memory, and tolerance. So the specificity piece is that antigens of pathogens will activate specific lymphocytes and the production of targeted antibodies. So the immune response will be specific to the antigen that has appeared in our body. That's the specificity. The versatility piece comes from the fact that you have an incredible diversity of lymphocytes. So you have millions of populations of lymphocytes, and each of those populations will be sensitive to different antigens. So hopefully when an antigen shows up in your body, you have a population of lymphocytes that have a receptor that will match that antigen. Now, your immune system also has ways to generate new lymphocytes with infinite varieties of receptors for antigens, but that's getting a little beyond the scope of this class. But let's suffice it to say we have a lot of versatility within the adaptive immunity system, but it sometimes can take time for that versatility to have an effect. We are going to have memory. So we will, in response to antigens, create cells that not only target that antigen to try to resist infection or illness and disease, but also to keep a memory of that particular antigen so that should we be exposed to it a second time, we can mount a much more effective and faster response to that antigen. And tolerance is another important property because tolerance means the immune system can not respond or ignore normal tissues and just target the abnormal and foreign cells in addition to toxins. So that's an important piece of adaptive immunity. We don't want to turn on adaptive immune responses in inappropriate situations. Now, how do we get adaptive immunity? Well, there's two ways. We can have active immunity or passive immunity. So active immunity will happen after exposure to an antigen. You might be exposed to that antigen naturally, as in maybe you were hanging out with someone that had that pathogen, and if it was an airborne illness, maybe they coughed or sneezed in your vicinity and you breathed it in. So you have now acquired that pathogen and it will trigger an immune response. Or you might be exposed to an antigen through vaccines, right? Vaccines are these incredibly safe and highly effective way of exposing our immune system to antigens of diseases or illnesses so that we can create an immune response to those specific antigens and have those memory cells. So then if we are ever exposed to that pathogen in the future, for example, measles or polio, then we will be able to mount a very rapid, effective response. Oftentimes, we won't even know that we were sick or infected. Or if we do develop illness from exposure to the antigen, we are much less likely to get seriously ill or require hospitalization. So these are an incredibly strong and effective way to promote public health through artificial acquired active immunity. Now, the other way that you might acquire immunity is through passive immunity. So this is when antibodies that have been made in response to an antigen are transferred from one individual or maybe the lab to another individual. Or from mom to infant, for example. So, when you naturally acquire passive immunity, two ways that might happen is across the placenta during fetal development. So, mom's antibodies pass through the placenta and into baby, or through breast milk. Both of these are highly effective means of protecting babies from potential diseases when their own immune systems are still developing. So they don't have a lot of natural immunity because they haven't been exposed to much. So they're reliant on their non-specific immunity, but the problem is the non-specific immunity isn't fail proof, right? So that's where this naturally acquired passive immunity is so great. Now, you can also artificially acquire passive immunity when we administer antibodies from, like I said, either from one person or if they're synthesized antibodies from the lab into an individual, and then those antibodies can target a particular antigen or pathogen. Antibody treatment was used during COVID for some high risk individuals. I wish I'd looked up more about it before doing this video lecture. I think it was pretty effective for some people, but there was also a real variety of efficacy. So, in other people it wasn't as effective, and that may just be because it was a relatively new disease. We're still, the science on antibody and cloning antibodies for use is still, I wouldn't call it new, but we're not old school at it yet. All right. Okay. So which type of acquired immunity develops after a vaccination? Pause the video if you need time to read. And there we are. With vaccination, it would be artificially induced active immunity. So it's active because you actually were exposed to the antigen and mounted an immune response, but it's artificial because it was through vaccination. All right, so now when we think about adaptive immunity, we want to sort of think about two different types of adaptive immunity. So adaptive immunity, big picture, is when antigen presentation triggers an immune response. And remember, antigens are just pieces of pathogens or toxins. They're often proteins. They don't absolutely have to be proteins, but they almost always are. And so we refer to the antigen as what we present because it's hard to present an entire pathogen. They're too big, whereas antigen is a little unique piece of that pathogen. So adaptive immunity, presentation of the antigen triggers an immune response. Now, the two responses that we can get are the cell-mediated immunity. This is going to be mostly involving T cells. And then the other type is antibody-mediated immunity. This is going to involve B cells. So in T cell activation with the cell-mediated immunity, what we're going to see is that once T cells are activated because they have been presented with antigen, those activated T cells will find the pathogens and destroy them either through phagocytosis or through the release of chemical toxins. And we'll go into a little bit more detail on that. In antibody-mediated immunity, this is where B cells that are activated through antigen presentation are going to produce cells that will give rise to antibodies that target that particular antigen. So when B cells are activated, they become plasma cells, and those plasma cells will produce antibodies, and those antibodies will go and bind to the antigen and help to destroy it. So we are going to talk about both of those in a little bit more detail. So first and foremost, what is this antigen presentation I'm talking about? How do we in fact trigger adaptive immunity? Well, it turns out that antigens need to be presented. They can't just be like floating around in the body. No, they have to be presented on like a little stage, basically. And that stage is called an MHC protein, which stands for major histocompatibility complex protein. Just call it MHC. All right, so basically, antigens have to be presented on an MHC protein, and that MHC protein could be on the surface of an infected cell or on the surface of a phagocyte. So one way or the other, In order for your immune system to see an antigen, I just put finger quotes around the word see, an antigen, it has to be presented to them on this MHC protein. All right. That MHC protein comes in two different types that we care about. There's the MHC1 protein and the MHC2 protein. They both present antigens, but the difference is which cells have which MHC protein. So all nucleated cells, any cell with a nucleus, with a couple of exceptions that I'm going to talk about, but all nucleated cells will have MHC1. So if an ordinary, everyday nucleated cell gets infected with a virus, it could take an antigen from that virus and present it on the surface of its cell membrane using MHC1. But there's another type of MHC protein called MHC2, and only certain cells have MHC2. Those are going to be any cell called an antigen presenting cell. Those will include monocytes, macrophages, and dendritic cells. Dendritic cells are those Langerhans cells that you saw in AP1 in the skin, if you remember that. And all the lymphocytes, like B's and T cells, will have MHC2. So kind of maybe like not a perfect rule, but a general rule is like the cells that are doing the work of the immune system, they're MHC2 protein cells, whereas all the other cells everywhere else in your body are going to use MHC1. And the reason why this is important is because it lets other cells know information. So if an antigen is on MHC1, then your immune cells will know that that's an infected cell showcasing that antigen. Whereas if it's on an MHC2 protein, then any other cell will know that that's an immune cell that's presenting it. So it just helps connect cells with the right cells and sending the right message about what needs to be done. All right, so class 2 MHC proteins are only present in the membranes of which of the following? So pause the video if you need time to read. And there we go. We're going to find MHC2 proteins only on antigen-presenting cells and lymphocytes. All the other kinds of cells are going to use MHC1. All right, so what does this look like? Well, let's take this image from your book and walk through it. So in this case, we have an infected cell. I know that because it says infected cell right here. But I also know that because I can see that this cell is using an MHC1 protein. And if it was MHC2, then I would know it was an immune cell or an antigen presenting cell. So therefore, I know this must just be a regular old infected cell since it's using MHC1. So here's what's happened. We've had a virus or a bacterial infection of this cell, and that has triggered antigen presentation. So in other words, our cell knows that it's been infected, and it wants to try to give a warning to other cells and show the immune system like, hey man, I've been infected, and this is what I've been infected with. So how that's going to work is that the infection actually creates the appearance of abnormal peptides. Remember, that's little short amino acids or little short like small proteins in the cytoplasm, which in turn will cause the endoplasmic reticulum to produce those class 1 MHC proteins. And then those little bits of protein from the infective agent, whether it's a virus or a bacteria, will be attached in the to the MHC1, and then that will be sent to the Golgi, and then the Golgi apparatus will package it up in a vesicle and send it up to the surface of the cell. And so now our infected cell is displaying our antigen from the infectious agent on an MHC1 protein on the surface of the cell membrane. So now other cells are going to be able to come along and be like, whoa, this cell is infected. And I know because it's showing me an antigen on an MHC1 protein. So it conveys a lot of information. Now, in this image, what we're seeing is what happens when an antigen presenting cell engulfs a pathogen. So this could be a cell that just goes around and chews up any kind of foreign anything that it finds. So it wasn't targeting this particular pathogen, it just happened to find it. So this could be a phagocytic cell that works within the innate immune system. So it engulfs this pathogen, and then a lysosome merges with the vesicle with the pathogen in it and starts to break it down into little bits. And that's when the little bits can be bound, the little protein bits can be bound to an MHC2 protein, and that will move out to the cell membrane and be displayed on the surface. So now our macrophage or our monocyte or whatever this cell might be is now showcasing an antigen on an MHC2 protein, which means it can alert other immune cells to the presence of this pathogen. And the other immune cells will be like, oh, this is my fellow coworker who discovered this pathogen, and now they're showing me the antigen that I should be on the lookout for. So that's another way that this specific immunity could be triggered. So here's an image of an actual antigen presenting cell and a lymphocyte. So the yellow areas are where we have MHC2 and antigen binding. And so the antigen presenting cell is presenting the antigen to this lymphocyte. And so therefore, this lymphocyte must have a specific receptor for whatever antigen is being displayed. In other words, this is the population of lymphocytes that will uniquely match with whatever antigen this APC is presenting. And I just want to be clear that that is just due to chance, that you have this system where you make millions and millions of lymphocytes, and each of them have variable receptors in the hope, or I don't know if it's hope is the right word, but on the chance that they match up with an antigen of a pathogen that you never knew you were going to be exposed to. I mean, it's really amazing. If you think too much about it, you'll start to just be like, oh my God, it's unbelievable. All right, so here's a quick question. Pause the video if you need time to read it. And these would be antigen presenting cells. Okay, so how do we specifically activate T cells? So how do we trigger them, and what do they do once they're triggered? Well, first and foremost, inactive T cells can respond to antigens on both MHC1 and 2. So T cells are capable of responding to antigens that are presented to them by regular old infected cells. So they're showing the antigen on MHC1, but inactive T cells can also respond when other antigen presenting cells of the immune system show them an antigen on MHC2. So inactive T cells can respond to antigens on either one of the MHC proteins. And if they match, if the receptor on the T cell matches the antigen that's being presented, then we get antigen recognition. And at that point, we need to know a little bit more information about T cells. So T cells actually have some special proteins that are going to help with that recognition. They are called CD markers, and there's actually a whole bunch of them, but we're just going to worry about two of them, which are CD8 and CD4. So CD8 is found on two kinds of T cells, cytotoxic T cells and regulatory T cells, and they are capable of responding to antigens that are presented on MHC1. In order for a T cell to respond to antigens presented on MHC2, they're going to need a CD4 marker, and the only T cells that have the CD4 marker are helper T cells. So when we say inactive T cells can respond to either MHC1 or MHC2, That's a true statement, but we have to remember that there are different kinds of T cells. And within the different kinds, some of them will respond to MHC1 only, and some will only respond to MHC2. So CD8 is cytotoxic T and regulatory T cells. CD4 is helper T cells, and they will respond only to MHC2. And that's going to be important, and you'll see why. All right, so now the question becomes, what happens if we activate CD8? So if we have a T cell, an inactive T cell, that has the CD8 marker, then it could respond to an infected cell that's displaying an antigen on MHC1. And if it does, that means we have activated the CD8 T cell. So that's where it gets confusing, is that a cytotoxic T cell and CD8 T cell is the same thing for us. So if we activate that T cell and it's a cytotoxic T cell, then that means it's also got the CD8 marker on it. So it's also known as a CD8 T cell. When we activate it, because it recognizes the antigen, that will lead to cell division. And if that cell undergoes cell division, it's going to create identical copies of itself. So whatever receptor it had that matched that antigen, it's going to make a whole bunch more cells with that same receptor. Some of those cells will be more cytotoxic T cells that will immediately go to work trying to find that antigen everywhere in your body and destroy it. Some of the cells, the daughter cells, will be memory T cells. They're going to just go hang out somewhere, like maybe in a lymph node or a malt patch. And they're just going to hang out in case you're ever exposed to that antigen again. And then they can be activated really quickly to make lots more cytotoxic T cells. And then we'll also make regulatory T cells, because at some point we got to shut this whole response down, because once we deal with the infection, we don't want the T cells, the cytotoxic T cells, to keep, attacking things, right? We just want to deal with the current infection, and then we want to tamp the whole thing down. So, how do cytotoxic T cells destroy the antigen? Well, they're going to go hunt down anything with that antigen. So it might, let's say it's a virus or maybe it's a bacterial cell. Every time they find that antigen, they will destroy the target cell. And they can do that in a bunch of ways. They can use a chemical called perforin, which will create holes and lyse open the cell. They can use chemical signals called cytokines, and they can also use lymphotoxin. So they have multiple weapons in their arsenal to destroy the target cell. Another thing is something called co-stimulation. So this is a process that we don't get super into detail on, but I want you to know about it, because co-stimulation is necessary to go from recognition of the antigen to actual activation of the T cells. And it's sort of it's like an extra layer of protection to make sure that your immune system doesn't respond to things that it shouldn't be responding to. Now, is it perfect? No, that's why we have allergies. But on the other hand, hopefully, we're not responding to absolutely every single thing in our environment, even if it's not dangerous. And that's part of co-stimulation. It's just making sure that whatever this antigen is actually like really bad for us. Super important to remember that the first time we're exposed to something, this process takes time. Just having the T cell with a matching receptor find the antigen and discover the match, that can take a lot of time. You got millions and millions of different T cells with different receptors, and the needle in the haystack has to find the antigen that matches. But the second time we're exposed to it, it's so much faster, because you've got all those memory cells hanging out in different places in the body. So they're more likely to spot that antigen, and then they respond really quickly. And again, you'll probably never even know you were infected, because they'll shut that down really quickly. And that's, again, one of the advantages of vaccines, is they give us those memory cells to be able to fight off subsequent exposure. All right, So what does that look like in pictures? Well, here we are. So here's our infected cell. So since it's an infected cell, that means it's going to display antigen on MHC1, but CD8 T cells, these are inactive T cells, they have CD8, so they can respond to antigen on MHC1. So that's what we're seeing here in this little blow up of the picture. So there's our T cell receptor, there's the antigen on our class 1 MHC protein being displayed by our infected cell. And then there's our CD8 protein that's hanging off of our CD8 T cell. And this is what allows this CD8 T cell to recognize antigens on MHC1. And then also, like I said, we're going to have this co-stimulation. Now, co-stimulation, again, I don't want to get into this in this class too deep, but sometimes co-stimulation is when there's an actual like ligand and receptor binding between two cells. And sometimes it's a series of chemical signals exchanged between the two cells. But either way, it means there's like a confirmation that this is like a for realsy situation that we should address and do something about. So if we get this business, then our CD8 T cell will become activated and it will undergo mitosis to create genetically identical daughter cells. Some of those daughter cells will become active cytotoxic T cells that will go look for the target pathogen that has that antigen. And if it can, anytime it finds that antigen on the target cell, it will destroy that target cell. It can do that with perforin, which will result in openings in the target cell, which will lead to lysis. It can use cytokines, which is a chemical signal that causes the target cell to undergo apoptosis, which means it kills itself. Or it can use a lymphotoxin release, and lymphotoxin disrupts cell metabolism. So basically it throws off the pathogen cell. It's not able to do what it needs to do to produce like ATP and other important enzymes. And so the target cell dies as a result. So that's the cytotoxic T cell. We're also going to produce memory cells. So those will be inactive cytotoxic T cells that could be reactivated if that antigen is presented to them again in the future. And it would be much faster that second time around. And then we'll also have those regulatory T cells that are a little slow to get going. But once they get going, they will help suppress this immune response so that we don't maintain it indefinitely. Again, once we destroy the target pathogen, we don't want to keep activating cytotoxic T cells and keep making memory T cells, right? We want to, like, once we do it once and we're good, then we need to just shut that whole cascade of events down. Okay, so I mentioned on an earlier slide helper T cells. So how are we going to activate these cells? And it turns out that these cells are really important because they are also going to help us activate B cells, which are responsible for that other type of adaptive immunity. So to activate CD4 T cells or helper T cells, they are going to need to recognize antigens presented on MHC2. So remember, these are the cells that have the CD4 marker, so they only respond to antigens presented on MHC2. So basically, when other immune cells, antigen-presenting cells, or other lymphocytes show them an antigen on MHC2, that's how they get activated. When CD4 T cells are activated, they will divide to produce active helper T cells and, of course, memory helper T cells. They do a bunch of things. They may bind to antigens, to their antibodies, which leads to sensitization. That just means that they are going to help increase the likelihood of endocytosis of the antigen. And sensitized B cells will need a helper T cell in order to release cytokines that in turn will activate the B cell. So this is sort of a complicated series of events in order to get our B cells activated. But once they are activated, then that B cell will undergo cell division to form plasma cells and memory cells. So let's just take a look in pictures because I think it's easier to understand in pictures. So here is an APC cell. So this is an immune cell of some type, maybe a macrophage or a monocyte. It has found an antigen and it is displaying that antigen on MHC2. And MHC2 is recognizable by antigens on MHC2 are recognizable by CD4 cells that are inactive. So a little bit of a blow up here of this picture. Let me see if I can get it a little bigger. Here we go. So there's our MHC2 protein on our APC. There's the antigen that would be displayed by MHC2. And then here on our inactive CD4 helper T cell is a receptor. And if it matches that antigen, that is the beginning of the process of activating this inactive helper T cell. You can also see the CD4 protein here. And so the CD4 protein is how we know this is a CD4 cell that will respond to MHC2. And then also we're going to have some kind of co-stimulation, just confirming that all of this is, correct and requires a response. And so if we get that co-stimulation, then what will happen is we will begin to activate this CD4 helper T cell. But you'll see, so there's our cell here, and now it will become active, and the active helper T cells will give off cytokines, and those cytokines will be important for activating additional immune cells. And then also we will have memory helper T cells. These are also inactive, but they're going to go hang out like in lymph nodes and other places like that in case we're exposed to this particular antigen again. Now, there's a little bit more to this. So that sensitization process that I was talking about. So once we have antigens on an inactive B cell, so they're binding on an inactive B cell to antibodies, right? These are the matching antibodies that the B cell has. So the antigen is binding. That helps to sensitize the B cell, because what will happen is the B cell will see, the antigen that binds to its antibody, it will bring into the cell and then display it on its MHC2 protein. And when that happens, then that sensitized B cell can bind to an active helper T cell. So we've already got that active helper T cell from earlier. That induces that response of cytokines, and then those cytokines act as co-stimulation between the helper T cell and the sensitized B cell, further stimulating the division of helper T cells. Let me try that again. The co-stimulation of the active helper T cell will further stimulate and activate the B cell to develop into a plasma cell to produce antibodies. So here's kind of the next steps there. So here is our active helper T cell, our CD4 T cell. It activated a B cell, a sensitized B cell, right? So it activated that. That underwent mitosis to produce memory B cells, and then also underwent cell division to create active B cells, which become plasma cells. Plasma cells then produce the antibodies that will travel throughout the body and bind anytime it sees that antigen. Okay, so here's a quick question. Pause your video if you need time to read it. B cells are primarily activated by activities of helper T cells. Absolutely. How about this one? Upon subsequent exposure to injuries or infections that induce the same antigen, memory B cells respond by differentiating into, pause if you need time to read, plasma cells, because B cells, when activated, can either become memory B cells or plasma cells. If you already have memory B cells, so it's a second exposure, then they are going to differentiate into plasma cells to immediately start producing antibodies and fighting off that infection. All right, so in this video lecture, we talked about adaptive immunity, which is highly specific and targets a particular pathogen through antigens that are unique to that pathogen. We have two major pathways for that. We are going to have our cell mediated immunity, which is mostly governed by T cells, and then we're going to have the antibody mediated immunity, which is governed by B cells. Again, both of these systems work together, and cell mediated immunity with T cells is going to help activate B cells. We cannot activate B cells without active helper T cells. So that's an important piece, because if something were to happen to your helper T cells where they couldn't be activated, then you couldn't turn on the antibody mediated immune response, and you'd have no way to produce antibodies. And that's basically what HIV does. It targets helper T cells. And so when those helper T cells are all shut down and you don't have any working, then you can't activate B cells. So that's a problem, because then you can't produce antibodies. All right, so I think it's a really good idea to walk through these diagrams in your chapter, and focus on what are the different types of cells and what activates each type of cell and what do they do once they're activated. So learn it in like little chunks, I think, is the easiest way to approach the immune system.